Okay, good morning everybody. So we are talking about the cells, right? Cell is the structural and functional unit of the living body or organism. In last class we talked about the structure of a cell. We talked about the structure of a cell membrane, the functions of different organelles, right? Functions of RNA. We talked about the proteins in the cell membrane. Two types of proteins, you know that, integral and peripheral. And we talked about the important functions of those proteins. So those are the main things we discussed in last lecture. Today, first we'll talk about the cellular junctions, also called intercellular junctions or joinings. Cellular junctions hold the cells together. If you see an epithelial tissue, you have already seen that, like these are columnar cells, and the cells are attached to each other. We always draw like this. You must remember flat cells like this. So these cells are attached to each other here, and these are the intercellular junctions or joinings. Okay, so adjacent cells are attached together by those junctions. So those junctions are present in the cell membrane of the adjacent cells. Three types of junctions are most common in the body. What are those three? Tight junctions, desmosomes, and gap junctions. So we'll see how they look like. Tight junctions don't have pores. So the small molecules, even small molecules cannot, or fluid cannot pass through the tight junctions. So the fluid or molecules from one cell to another cell cannot pass through the tight junctions. Makes sense. Name is tight, right? So tight junctions. Desmosomes. Desmosomes are very strong junctions. They hold the cell membranes very strongly like this. Look at me. Protein structures come out from the cell membranes, adjacent cell membranes, and hold the cell membranes very strongly like this. It is like zipper in your clothes. You know, you use zipper very strongly, hold the cell membranes together. So, you know, sometimes when your skin gets dry, you can peel the skin. A chunk of skin comes up, right? You have seen snake. When they change the skin, the whole thing comes out together. Why? Because the cells are held strongly by desmosomes. In your heart, you know that whole muscle, heart, should contract together, right? All muscle cells should contract together. So they are strongly held by desmosomes. Make sense? So they will work together. So in some areas of your body, you have plenty of desmosomes. For example, skin, heart muscle. Gap junctions. Its name is telling you. it is not tight. That means through gap junctions you have pores or holes. So small pores or holes are present. So the fluid and molecules can easily go from one cell to the next. Okay? So the ions, molecules, fluid can easily move from one cell to another. Make sense? So a lot of pores or holes are present in the gap junctions. So those are three types of junctions, and they are formed by peripheral proteins, because you know when we talk about the functions of the peripheral proteins, one important function is intercellular joining. Intercellular joining is intercellular junctions. Okay? So basically the peripheral proteins form these intercellular joinings or junctions. Okay? So here you see, first let's see the structure of a tight junction. You see the tight junctions are here. So adjacent cells are attached by tight junctions, and if you see the structure under microscope, you will see this is one cell membrane, this is the other cell membrane, and they are attached to each other by these proteins, peripheral proteins. Okay, these are called interlocking junctional proteins, one type of peripheral proteins, and it's like, you know, two sheets of clothes you take and stitch. Make sense? Together, sometimes you do that, right? Two pieces of clothes, then you stitch. So the protein molecules, peripheral proteins, are stitching them together. Desmosome. This is one cell membrane, this is another, and you see in the inner side of the cell membrane you have protein structures like disc-shaped protein structure that is called a plaque. So this is another plaque, so it's like a disc, and from that plaque these interlocking proteins come out and they get attached to each other like this, I mentioned. Okay, so these are the proteins coming out, these are the proteins coming out, and hold them together. So these proteins are called linker glycoproteins or cadherins. Remember the name, I may ask you. So linker glycoproteins or cadherins, they come out from the plaques of adjacent cells and get attached to each other like zipper, like strong attachment. Okay, so you cannot separate the cells easily, they will stay together strongly. As I have mentioned, in your skin, heart muscle, you have plenty of desmosomes. Gap junctions have pores through channels. So you see, these are gap junctions, and if you see under microscope, you will see through these peripheral proteins, you have channels or pores. Okay, so the molecules, ions, fluid can easily move from one cell to another. This is very helpful. Because of the gap junctions, what happens, the ions can easily move from one cell to the next, which is very important. For example, muscle contraction. When your muscle contracts, right, all muscle fibers must contract together to make strong contraction, right? So they need to quickly talk to each other so they can do together. Make sense? So the exchange of ions can occur easily because of the gap junction. Make sense? So that is one example, that all muscle fibers when I contract must contract, and they need to talk to each other, and exchange of ions occur through the gap junctions. That is very important. Okay, plenty of gap junctions are present in the muscles, including your heart muscle. So in our heart muscle, if I ask you which two types of junctions are present, one is desmosome, that hold the membranes very strongly together like zipper, and another is plenty of gap junctions. So the cells can talk to each other quickly, okay, exchange ions quickly. Both are very important for the heart muscle to contract strongly. Okay, so these two are present in the heart muscle in huge amount. Okay, so the heart muscle fibers work together. Transport through the cell membrane. You know that cell membrane is a semi-permeable membrane. I mentioned in last class, right? Semi-permeable. That means what? Partially permeable. It allows certain chemicals pass through it. Not everything, right? If everything passes, we will say what? Fully permeable, right? If nothing passes, we will say impermeable, right? But I said semi-permeable, that means partially. It blocks some chemicals, but also allows some chemicals. So, partially permeable membrane. And it is very important that cell membrane is semi-permeable, because you know that inside the cell, a lot of activities, right, go on. And this is extracellular fluid outside of the cell. This is intercellular fluid, cytoplasm inside the cell. And this is the cell membrane. So certain chemicals should enter and certain chemicals should get out. Those chemicals, for example, oxygen, water, those are needed for the cell. Nutrients, oxygen, water, they will get in. Make sense? We eat food, right? Nutrients come from food and given to the cells, tissue, right? We drink water. Those should enter into the cell of your body. And also oxygen. Make sense? So those things should enter into the cell. And inside the cell, you know, cellular metabolism occurs. Right? Metabolism occurs inside the living cell. And when metabolism occurs, we know that ATP is produced, right? That gives energy, which is good, but also toxic chemicals, metabolic wastes are produced, right? Should they stay inside the cell or get out? They should get out, right? So you see that some chemicals should get in, some chemicals should get out through the cell membrane. So that's why it is semi-permeable. But remember, it should not let the toxic chemicals get in easily. Make sense? Toxic chemicals should get out, not in. If it gets in, it is harmful. It can kill the cell. So anyway, so that's why through the cell membrane, transport of chemicals take place. And that is called the membrane transport. Now, two types of transport occur through the cell membrane. One is called active transport, and another type is passive transport. In active transport, you need ATP or energy. Okay? ATP required. It is called active. You need energy. Make sense? Active. Like, you know, if I ask you to run and go to that corner of the room, you have to actively move, right? You need to use energy. Otherwise, you won't be able to move. Make sense? Now, if you sit on something, or I hold you like this and move you to that corner, you are not utilizing any energy, right? Make sense? So that is passive. So, in case of active, you need to utilize ATP or energy. In case of passive, no ATP or energy is required. So that's why active transport can only occur through the living cell membrane, because ATP is only produced by the living cells, produced in the living cell, right? By metabolism. Metabolism doesn't occur in non-living things. Make sense? So ATP is produced only in the living body. Make sense? And that's why active transport can occur only in the living body. Passive can occur in living as well as in non-living membrane, because no energy or ATP is required. Okay? In the lab, if I put two different types of solution and put a filter paper in between, then what will happen? Some molecules will go from this side to that side, right? Some will go from that side to this side. That is passive. No energy is required. So those are two types. Now, passive could be simple diffusion, facilitated diffusion, and osmosis. So those are three different types of passive transport. Let me explain you. So this is a cell membrane, semi-permeable membrane, and in this side, if you put high concentration glucose solution, so these are glucose and water. Okay, so this is glucose solution, and in this side you also have glucose solution but low concentration. So these are glucose molecules and plenty of water in this side. Less glucose, more water. Make sense? And in this side you have more glucose molecules because concentration is high in this side. Is it clear? You know that. So this is high, this is low. Now, in this situation, what will happen? Remember one thing which is very important: equilibrium must brought back. That means equilibrium should be maintained. Both sides should have, the goal is bringing both sides solution same concentration in both sides. Make sense? So just think that that is the goal, making both sides solution same concentration or equal concentration. Now you can easily tell me, in this side, what is less? Glucose is less, right? Make sense? So glucose should move from this side to that side to bring same concentration. And in this side, what is more? Water is more, right? Than this side. So water should move from low to high. Is it clear? So if the solids move, glucose moves from this side to this side, then concentration will go up gradually, and if water moves from this side to this side, concentration will go down gradually. Make sense? So finally, when in both sides the concentration will be equal, movement will be stopped. Make sense? Okay, so you see, two things are moving here. Solute is, or glucose, which is a solute, and water is a solvent. So you see, solute is moving from high concentration to low, and that is called osmosis. sorry, that is called simple diffusion. Simple diffusion. Okay, and water is moving from low to high. That is called osmosis. Okay, so simultaneously, simple diffusion and osmosis will occur. The goal is again bringing same concentration in both sides. So now you've got the idea what is simple diffusion, what is osmosis. In simple diffusion, who moves? Solute or solvent? Solutes from high to low. Very good. Makes sense? And in osmosis, who moves? Solute or solvent? Solvent from low to high. Make sense? Okay. Most of the cases, solvent is water. Because water is the most common, right, chemical in the body, and most of the chemicals are dissolved in water. So when we say solvent, in most of the cases we indicate water. Solutes could be many thousand different types. Okay? So now another thing is called facilitated diffusion. This is the cell membrane. Okay, you already know in the cell membrane you have proteins, integral proteins, peripheral proteins. Okay, so this is again solutes, this side solids. So this is the integral protein. In this case, what will happen, this glucose molecule will come and there is a binding site here like this. It will come in, get in, and will sit here like you are sitting in an elevator that will take you up. Okay, then what happens, this side will get more open like this, get more open, and this solute will be released into this side. So what happens, first the solute will get in, will sit on the chair here, and then that side will open like this and will release the molecule to the other side. So in this case you see that this integral protein is helping or facilitating this diffusion. Okay, so that's why. So it is moved by the help of something. Okay, so it is sitting on your shoulder, then you are taking the person to another place. Excellent. So first, someone is sitting on your shoulder, then you are moving and dropping him. Make sense? So that's the facilitated diffusion. It doesn't happen directly through the pores or channels. It will first get attached to the carrier, and then the carrier will take it to the other side. That's why it is also called carrier-mediated diffusion. You can test the simple diffusion and osmosis in the lab. As you know, those things happen in non-living membranes too. So you can put a membrane in between two solutions. One is high concentration, another is low concentration, and in this case you see the pore through this membrane is very large, larger than the size of the solute or sugar. So in this case the solutes will move from high to low, we already know, simple diffusion, and water will move from low to high, that is osmosis. So in this case both simple diffusion and osmosis will occur, and the goal is, you see this side, same concentration on both sides. Make sense? So establishing equilibrium. Now, you can only, let the water pass through but block the solids. So in this case, only osmosis occurs. You see, if the membrane has pores, but those pores are smaller than the size of the solutes. So in this case, solutes cannot move, but water or solvent will move from low to high. So what will happen? The volume of solution in this side will increase. Volume doesn't matter to move the molecules. The matter is the concentration difference. So since the concentration is high in this side, solutes cannot move, only solvent will move, and concentration, you see, is same in this case. This side and this side, the concentration is same. Although the volume is much more in this side. Make sense? So bringing concentration equal, that is the goal, right? Not the volume. So in this case, only osmosis has occurred, movement of water. Response of a living cell in different types of solution. If you put a living cell in isotonic, hypotonic and hypertonic solutions, how the living cell will respond. Now first you need to know what does mean hypotonic, hypertonic, isotonic. So let me show you. This is a solution. Okay. For example, again, glucose solution. Okay. Now, if you put a red blood cell or any living cell inside this, if the concentration of the cytoplasm here inside the cell is 5 milliosmol, that's the unit of concentration, okay? 5 milliosmol inside the cell. And outside of the cell, the concentration is 10 milliosmol. Now, very simple question. Compared to inside, outside is hyper or hypo? Hyper means what? More, right? Hypo means low. So I said outside is hyper or hypo?
Hyper.
Everybody.
Hyper.
Compare, I said compared to inside, outside is more or less? More. That means hyper, right? Okay. So, you know, water moves, solvent moves from what? Low to high in osmosis, right? Water always moves from low to high. Now you tell me which way water will move. Makes sense, right? From low concentration to high concentration. So if water comes out from this cell, the cell will get bigger or smaller?
Smaller.
Smaller, shrink, right? So that will happen in hypertonic solution. This is hyper. It is a relative concentration. We are comparing to hypertonic. Okay? So in hypertonic solution, water will come out from the cell and cell will do what? Smaller. Makes sense? Shrink. Shrinkage of cell. Now, let's change the concentration of solution. I put this cell in a different concentration solution. In this case, the concentration is 2 milliosmol. So now you tell me, the solution is hyper or hypo compared to this one?
Hypo.
Hypo. Make sense? Less. Water will always follow the same rule from low to high. Make sense? Osmosis. So in this case, what will happen? Water will move from low to high. Make sense? So water will get into the cell and cell will get what? Bigger. Make sense? So swelling of the cell, enlargement of the cell will occur. Eventually, it may rupture. Okay? If the cell membrane is not strong enough, make sense? It may rupture. So, that is the response in hypotonic solution. Now, iso. Iso means safe. Iso means safe. So, if the concentration between inside and outside, inside and outside, the concentration is same, then no need to move the water molecules. Make sense? Equilibrium is already there, right? So, no change of the cell. Is it clear? Okay. So, those are the responses of a living cell in 3 different types of solution. Now, you tell me, you are a nurse or doctor. If someone asks you to put solution because the person got dehydrated, okay? So you will put plenty of solution inside the vein, in the blood. So what kind of solution you will prefer? Do you want to bother the cell? No, you don't want to bother the cell, right? You don't want them to get big or small. So which kind of solution you will use? Isotonic. Make sense? Because you know that in hypertonic the cell will get shrink. In hypotonic the cell may even rupture, right? So you will use isotonic solution. Okay? We use 0.9% sodium chloride, which is the standard for the body. So same thing you see here, red blood cell and the response of it in 3 different solutions. Okay? Isotonic, no change. This is in hypertonic, so shrink, and in hypotonic to get big. Okay, awesome. Okay. Okay, so we want to start a new chapter because your first lecture exam is over orientation of the human body. Okay. Second chapter chemical, chemistry, right? Chemical level, and third one is the cellular level. Okay. I have posted some practice quizzes. I will post more, so keep practicing. Okay. And if you have any question, just come to my office, ask me. But practice. More you will go over. Okay. You'll see everything will get easy. If you think that I will study, just before the exam, that will not help. In anatomy and physiology, you have to keep you up to date, okay? And if you don't understand anything, feel free to talk to me, yes.
So our first exam is going to be over all 3 chapters?
3 chapters, yes.
Okay. When is it?
I don't know. You should know, you have the schedule, right? That's why I said you, get a printout of the schedule and put in your folder, so you don't have to come to me. When is my exam? You should take some responsibility. There is another class where we will do the practice things, okay? And then I believe. So today's Tuesday, Thursday, next Tuesday. But make sure check your schedule, okay? I'll check the schedule and follow it. I teach many sections, so I don't want to.
I have a question, with the practice.
Yeah, if you have a couple of questions, you can ask.
Oh, just to make sure that if we get the right definitions or the right,
These questions are straightforward questions. You should be able to find a PowerPoint in my lecture. I also post the lecture video, watch that. Everything is there. Okay, everything is there. If you find anything on yourself, still, if you have confusion, then don't have to fix it. I don't want you to look at what is the powerhouse of the cell. You should not need to. I have already mentioned. You should be able to know. Okay? So that's how I want you to learn. Okay? If you have a couple of questions, Then you can.
Sure.
Okay?